Please welcome Dr. Gail Besner as she addresses a pain in the neck in EC, research challenges and opportunities. Dr. Besner. You're welcome. Good stuff. Proud of you.
Well, thank you so much for that very kind introduction. Dr. Crummell, Dr. and Mrs. Grosfeld, members and guests. When Dr. Crummell called me months ago and invited me to deliver the 2014 Jay and Margie Grosfeld lecture, I was stunned. I immediately thought of the previous presenters of this address, each of whom gave powerful lectures, particularly recalling the 2013 lecture delivered by Dr. Jessica Candell. As I sat in the audience listening to that lecture, I vividly recall thinking that the 2014 lecturer would have impossibly big footsteps to follow. Gratefully, it never occurred to me at the time that it would be me, but here I am. I sincerely thank Dr. Crummell for the opportunity to share with you today a little bit about my research journey. Furthermore, I'm deeply indebted to Jay and Margie Grosfeld for the honor of delivering this lecture in their names, and we're all so grateful for everything that Dr. Grosfeld has done over the years to support APSA and the ability of young surgeons to perform surgical research. He's worked tirelessly, eliciting donations to the APSA Foundation, which go directly to support research enrichment scholarships, with previous award winners shown here. The APSA Foundation Scholarship was one of the first research awards that I ever received, and it was certainly instrumental in my ability to subsequently obtain additional external funding for my research. One thing that I can guarantee you is that if Dr. Grosfeld has not already approached you to donate, he'll do so soon, so be prepared to give. And as you can see from this list of award winners, the return of investment is quite good. The title of my talk today is A Pain in the Neck, Research Challenges and Opportunities. Please don't interpret my use of the term a pain in the neck as being flippant in any way. Rather, it reflects the pain that we all have to deal with when we operate on one of these small, fragile patients and have to tell the parents that we don't think that their baby will survive. The first case of neck that I ever operated on was during the 3rd month of my first year as a pediatric surgery fellow. Baby boy Freeman, a 900-gram preemie, survived the operation and lived, and I was elated. Today, so many years later, I hope to never have to operate on one of these babies again. The second half of my title, Research Challenges and Opportunities, reflects the ups and downs that we all face when doing research. As I prepared for this lecture, I reflected on what it was that actually instilled in me a passionate love for research. Could it be that the love for research is something one is born with? If you do a Google search of Besner in order to find the very first paper written under this name, you will not find any of my prior studies of growth factors, intestinal injury, or anything even written by me. You'll find this paper entitled The Law and Low-Level Radiation, which was presented at the Congress of the International Radiation Protection Association in Washington, D.C. in 1973. The purpose of the paper was to examine a number of latent radiation injury cases with emphasis on the kinds of radiation records offered in evidence, the nature of the expert testimony, and the conclusions of the court. You'll see that the very first case that was successfully won in court that linked repetitive exposures to low-dose radiation at the workplace with cancer, a New York case of an employee who was a theoretical physicist who died from acute myeloblastic leukemia, referred to as reference 30. Examination of the reference list shows that this case was Besner versus Walter Kitty Laboratories. Quoting from the paper, the record discloses that decedent was exposed to radiation for a substantial period, part of 2 periods, and also at other times in various amounts. The testimony of the medical experts is emphatic that there is really no threshold or safe dosage of radiation because at the present stage of scientific knowledge, it cannot be ascertained exactly what effects radiation has on the human body. The award is supported by substantial evidence, especially so in view of the decedent's good health prior to his employment. Well, none of you in the audience will recognize this man. This is Dr. Aaron Besner, who was born in 1923. He was a nuclear physicist who worked for Walter Kitty Nuclear Laboratories in New York City. His project was to design and build an atomic-powered locomotive. Those were in the days before the dangers of low-dose radiation were understood or able to be monitored. After radiation exposure in the research lab, he was diagnosed with acute myeloblastic leukemia in 1957 and died in 1958 at the age of 35. As you probably figured out, Aaron Besner was my father, who I never knew since he died when I was a baby. Although I never knew my father growing up, could he have instilled in me and left me with an innate love for research? I was raised by my mother, a single parent in the heart of New York City. I remember her telling me that science was in my genes. When I was in high school, I told her that I wanted to be a scientist. Having lost her husband to the field of science, I can only imagine what she must have thought. She was very supportive of my career decision, but told me that she thought I should consider doing research after first becoming a doctor, so that I would understand what problems patients have that really required research. Although I didn't understand it at the time, my mother, who had absolutely no medical knowledge, was describing a physician scientist. Despite never wanting to listen to anything that my mother told me to do, I took this piece of advice and I've never regretted it. I did my general surgery residency at the Brigham and Women's Hospital in Boston. Every year, Dr. Judah Folkman crossed over the bridge from Boston Children's Hospital to the Brigham to give a talk to the surgery residents. As soon as I heard him speak, I decided that no matter how long it took, I wanted to do research in his laboratory. Well, as you might imagine, many other people had the same idea. So it took me until after my 5th year of general surgery residency to get a research position in his lab, but it was well worth the wait. At that time in the Folkman lab, it was known that angiogenesis, or the growth of new blood vessels, was controlled by potent angiogenic growth factors such as basic fibroblast growth factor. However, basic FGF was known to be cell-associated and not secreted, so it was difficult to understand how it could go about exerting all of its potent angiogenic effects if it couldn't even get out of the cells. I was given a project to find a cell type that could naturally secrete basic FGF. Having done some research on macrophages as a college student, I immediately thought of looking at macrophages, since these cells are highly secretory in nature. Early every morning, I would arrive at the Dana-Farber Cancer Institute when they were spinning off the buffy coats from units of blood that had been donated. I brought these human white blood cells back to the lab, and from them I purified macrophages such as those shown here. After a year of working on these cells, I was convinced that I had done it. I had identified a heparin-binding growth factor that was naturally secreted from macrophages and that I thought was basic fibroblast growth factor. However, further studies showed that it had some properties that were different from FGF. During my 2nd year in the lab, it was sadly apparent that I had not identified a secreted form of FGF at all. However, further analysis showed that I had serendipitously discovered a totally new growth factor, which came to be known as heparin-binding EGF-like growth factor, or HB-EGF. This was the first report of HB-EGF that appeared in the literature, and that was in 1990. HB-EGF is a member of the epidermal growth factor family that's produced as a 208 amino acid precursor molecule that undergoes extracellular proteolytic cleavage to yield the mature secreted form of the growth factor. HB-EGF contains an EGF-like domain, as shown here in red, and also an N-terminal hydrophilic extension in which its ability to bind to heparin resides. And here we see the secondary and tertiary structures of the molecule that were subsequently elucidated. Well, since our initial description of HB-EGF in 1991, the number of HB-EGF publications worldwide has increased annually, and it includes reports showing that HB-EGF protects the brain from stroke, the heart from myocardial infarction, and the kidneys, bladder, and liver from different forms of injury. Over the last 2 decades, our laboratory has focused on studies showing that HB-EGF can protect the intestines from various forms of injury, including necrotizing enterocolitis. As pediatric surgeons, we're frequently confronted with the often devastating disease of neck. This illustration depicts the typical appearance of a baby with neck. These patients are typically born prematurely, and you can tell from the number of lines, tubes, and drains that are in place that the baby is quite ill. We see several loops of bowel that are significantly injured, and if you examined that intestine closely, you might see little bubbles of air trapped in the bowel wall, known as pneumatosis intestinalis. Despite the most advanced methods of caring for these patients, even including surgical removal of the diseased intestines, and despite over 6 decades of research, much of which has been done by several brilliant investigators right here in this audience, the mortality of neck remains unchanged and is as high as 50%. Sadly, neck is still a disease for which there's currently no known cure. This is the cover of the June 2nd, 2014 New York Times Magazine with the feature article titled "Saving Preemies." Contrast that with these images from a 1939 Life magazine article titled "Incubator Rooms Save Premature Babies' Lives," a time when weak babies were placed into the HES bed to give them air and oxygen to facilitate breathing. Newborn care units looked vastly different from the way they look today, and mothers had to view their babies through a glass partition since they were not even allowed to enter the unit. Compared to the progress that we've made in most other diseases, our progress on necrotizing enterocolitis, the leading cause of death in NICUs today, has been painfully slow. Over the past 2 decades, our laboratory has used multiple different animal models to examine the effects of HB-EGF on the intestines, including animal models of necrotizing enterocolitis, intestinal ischemia-reperfusion injury, hemorrhagic shock and resuscitation, cecal ligation and perforation, radiation therapy-induced intestinal injury, and intestinal anastomotic wound healing. And we've shown that HB-EGF protects the intestines from injury in the first 5 models and promotes wound healing in the 6th model. This is what our rat pup model of neck looks like. Rat pups are delivered by C-section prior to term, and starting shortly after birth, they're exposed to hypoxia in a hypoxic chamber, followed by hypothermia, and with the addition of hypertonic feeds and lipopolysaccharide, we see an appearance of the intestine that's very reminiscent of the clinical disease of neck that we see in the operating room. When HB-EGF is administered to these pups, we simply add it to the feeds that the animals receive, and we can perform this model in newborn mice in addition to newborn rats. This slide depicts the effect of HB-EGF in our neck model as seen by scanning electron microscopy. We see the appearance of normal small intestinal villi and breastfed pups on the left, compared to the severely damaged villi and pups subjected to experimental neck in the middle panel. Note that in pups subjected to experimental neck but treated with HB-EGF simply added to their feeds, there's significant preservation of the villus microarchitecture as shown on the right. The lower panels show higher power magnification, and note that there are numerous bacteria attached to the mucosa of the neck-injured villi, a pattern that was never observed in either breastfed pups or in pups subjected to neck but treated with HB-EGF. Over the years, we've conducted numerous lines of investigation examining the mechanisms by which HB-EGF protects the intestines from injury. We showed that HB-EGF promotes intestinal epithelial cell proliferation and migration and decreases intestinal epithelial cell apoptosis, all very important to promote wound healing in the intestines. Furthermore, HB-EGF decreases NF-kappa B activation, leading to decreased pro-inflammatory cytokine production, and it decreases inducible nitric oxide synthase expression, leading to decreased overproduction of injurious levels of nitric oxide. We showed that HB-EGF decreases neutrophil endothelial cell interactions, and it decreases the infiltration of macrophages and neutrophils into injured intestine. In addition, HB-EGF decreases reactive oxygen species production. On the other hand, HB-EGF increases endothelial nitric oxide synthase expression and acts as a potent vasodilator that promotes villous microvascular blood flow. Importantly, we showed that HB-EGF preserves gut barrier function and decreases bacterial translocation after intestinal injury. Well, recently we've become very interested in the effect of HB-EGF on intestinal stem cells, which are probably the most important cell type in the crypt-villus axis. This video, produced by Dr. Hans Clevers in the Netherlands, demonstrates the events that are continuously occurring in the intestinal mucosa. You can see that at the very bottom of the crypts are where these important intestinal stem cells are located. And in this case, they've been stained in blue so that you can appreciate how they proliferate and where they end up going. So here you can see those intestinal stem cells. They're proliferating. They're forming something called the transit amplifying compartment, and then they march up the crypt-villus axis, where they differentiate into all of the different intestinal epithelial cell lineages, including the mucus-producing goblet cells, the absorptive enterocytes, etc. Those cells then will continue to march up the crypt-villus axis, and when they finally get to the tips of the villi, they'll undergo the process of programmed cell death or apoptosis. You'll see that now. And that's how homeostasis of the entire crypt-villus axis is maintained. So here are the intestinal stem cells. There are those blue-green cells at the bottom of the crypts. And they go on to proliferate and to differentiate into all of the other intestinal epithelial cell lineages, such as mucus-producing goblet cells, hormone-producing enteroendocrine cells, and absorptive enterocytes, all of which march up the crypt-villus axis, as well as antibacterial protein-producing Paneth cells, which move down to the crypts to reside between those important stem cells. In this experiment, we utilized our experimental neck model to examine the effects of HB-EGF on intestinal stem cells of newborn mouse pups. To identify the intestinal stem cells specifically, we utilized LGR5 EGFP transgenic mice. These mice have fluorescently labeled stem cells so that we can easily visualize and see where the stem cells are. Pups exposed to NEC had easily identified, pups that were breastfed, normal pups, had easily identified stem cells. Pups exposed to experimental NEC had significant loss of stem cells, whereas pups exposed to NEC but treated with HB-EGF added to the feeds had significant preservation of their stem cells. Thus, the ability of HB-EGF to protect stem cells from injury may represent a key factor in its ability to exert all of its subsequent protective effects. Once we showed that HB-EGF could protect resident intestinal stem cells from injury during NEC, we performed a series of studies that demonstrated that HB-EGF could protect mesenchymal stem cells from injury in vitro. And based on those results, we hypothesized that administration of HB-EGF in conjunction with exogenously administered mesenchymal stem cells might further increase protection of the intestines from injury. Several papers resulted from our studies of the effects of HB-EGF and mesenchymal stem cells in both NEC and intestinal ischemia-reperfusion injury. In our model of NEC, we found that bone marrow-derived mesenchymal stem cells administered either IV or intraperitoneally preferentially engraft into injured intestine, and that simultaneous administration of HB-EGF plus stem cells decreases histologic injury, preserves gut barrier function, and improves survival. Using our model of intestinal ischemia-reperfusion injury, we found that both bone marrow and amniotic fluid-derived mesenchymal stem cells preferentially engraft into injured intestine, and we found that overexpression of HB-EGF in amniotic fluid mesenchymal stem cells is as good or better than administering both therapies together. Well, more recently, we've been examining the effects of HB-EGF on the enteric nervous system. As you know, the ENS is located in the wall of the intestine, is the largest and most complex division of the peripheral nervous system, and is responsible for intestinal motility, absorption, and secretion. It consists of a collection of ganglia containing neurons and glial cells. Whereas the neurons are responsible for GI motility, the role of the glial cells is less well understood. However, it is known that glial cells contribute to postnatal neurogenesis, or the growth of new neurons. And interestingly, ablation of glial cells leads to fulminant and fatal intestinal inflammation similar to the histopathology that we see in NEC. For the next few minutes, I'd like you to consider a new potential contributing factor for the development of necrotizing enterocolitis. Consider the possibility that enteric nervous system immaturity in premature babies may lead to poor intestinal motility, increasing vulnerability to NEC. Furthermore, we know from our experience that intestinal dysmotility is common after NEC and suggests that the ENS is injured during NEC and not fully recovered, even after survival from the acute disease. We began our ENS studies by investigating whether the enteric nervous system is in fact damaged during clinical NEC. To examine the ENS, we collected intestinal tissue specimens from 8 control patients with small bowel atresia and 18 age-matched patients with acute necrotizing enterocolitis. All patients underwent initial bowel resection with ostomy creation, followed by subsequent stoma closure 2 to 3 months later. We first investigated whether the ENS was damaged during acute NEC, and what we're looking at in these images are individual ganglia of the myenteric plexus, as shown in the red box. We see representative images from a premature baby with small bowel atresia in the upper panels compared to a premature baby with necrotizing enterocolitis in the lower panels. Compared to control intestine, we found that patients with acute NEC have significantly decreased neurons, significantly decreased glial cells, and significantly increased apoptotic cells in the enteric nervous system. We next examined the enteric nervous system from the same patients 2 to 3 months later at the time of stoma closure. We stained these sections for neurons and glial cells as before, but we also stained them for neuronal nitric oxide synthase, since its product, the neurotransmitter nitric oxide, is responsible in part for intestinal motility. Although the total number of neurons was similar, we found that NEC patients have significantly decreased glial cells and significantly decreased nNOS-producing neurons at the time of stoma closure, suggesting that the enteric nervous system has still not recovered from the initial NEC insult. We therefore confirmed that the enteric nervous system was injured during acute NEC, and it was not fully recovered even after survival from the acute disease. Similar to the central nervous system, the ENS is markedly restricted in its ability to regenerate damaged neurons. Others have shown that HB-EGF protects cerebral neurons in animal models of stroke, and we've shown that HB-EGF protects enteric neurons from injury in vitro. Based on these observations, we wondered whether HB-EGF could protect the enteric nervous system during NEC. To examine this, we went back to our animal model of NEC. In these images, rather than looking at individual ganglia as before, we're now looking at the entire myenteric plexus as shown in the diagram above. We see the myenteric plexus from breastfed animals in the upper row, from animals exposed to NEC in the middle row, and from animals exposed to NEC but treated with HB-EGF added to the feeds in the lower row. And below, we see the corresponding quantification of Western blots. In the first panel, we see normal neuronal protein levels in breastfed pups, significantly decreased neuronal levels in pups exposed to NEC, and significantly increased levels in pups exposed to NEC but treated with HB-EGF added to the feeds. And we found similar results for glial cells and for those important nNOS-producing neurons. Thus, it appears that enteral administration of HB-EGF can, in fact, protect the enteric nervous system during NEC. Well, since enteric neural stem cells are the cells that differentiate into the neurons and the glial cells that comprise the enteric nervous system, we next hypothesized that administration of HB-EGF in conjunction with neural stem cells or overexpression of HB-EGF in neural stem cells might further protect the intestines from NEC. To examine this, enteric neural stem cells were harvested from the intestines of pan-EGFP mouse embryos so that the cells are fluorescently labeled and we can track where they go. The cells were then grown in culture where they form neurospheres, and nestin staining of those neurospheres confirmed the presence of large numbers of neural stem cells. These neural stem cells were then transfected with either an HB-EGF plasmid so that they produce very high levels of endogenous HB-EGF, or with a scrambled DNA control plasmid so that they produce normal levels of HB-EGF. The cells were then administered intraperitoneally, and the pups then exposed to experimental NEC. Intestinal injury was graded using a standardized intestinal injury scoring system, in which grade 0 is normal intestine, grade 1 is epithelial cell lifting at the villus tips, grade 2 is necrosis to the midvillus level, grade 3 is necrosis of the entire villus, and grade 4 is transmural necrosis. And we consider any grade of injury of 2 or above as being consistent with necrotizing enterocolitis. Here we see our histologic injury results. In this graph, each dot represents an individual mouse pup. Pups that were breastfed had no neck. Pups exposed to experimental NEC had significantly increased intestinal injury. Pups exposed to NEC but treated with either HB-EGF alone, either neural stem cells alone, HB-EGF alone, HB-EGF plus neural stem cells, scrambled transfected neural stem cells, or HB-EGF overexpressing stem cells, all had a significantly decreased incidence of NEC, with the lowest incidence of NEC seen in pups treated with either HB-EGF plus neural stem cells or with HB-EGF overexpressing neural stem cells. Intestinal permeability was measured as the serum concentration of fitsidextran that was administered enterally 4 hours prior to sacrifice. The lower the serum levels in this experiment, the better the gut barrier function. Pups that were breastfed had very low intestinal permeability. Pups exposed to experimental NEC had significantly increased permeability. Pups exposed to NEC in all of the treatment groups had significantly decreased intestinal permeability, with the best gut barrier function seen in pups treated with a combination of HB-EGF plus neural stem cells or with HB-EGF overexpressing neural stem cells. In the next few experiments, we modified our rat neck model slightly such that after the regular 3 days of stress, we then recovered the animals for an additional 3 days, during which time stress was stopped and the pups were fed regular formula to mimic recovery or resuscitation from NEC. Here we see images of the enteric nervous system upon recovery from experimental NEC. The sections were stained with HUCD to show mature neurons in red, and for EGFP to show the labeled transplanted neural stem cells. Here we see the normal enteric nervous system in breastfed pups. In pups exposed to NEC, the number of mature neurons was significantly decreased. In pups that were exposed to NEC but treated with neural stem cells, we see the green transplanted neural stem cells, some of which have differentiated into mature neurons. And in pups exposed to NEC that were treated with HB-EGF overexpressing neural stem cells, the numbers of mature neurons were further increased. Therefore, HB-EGF enhances the engraftment of transplanted neural stem cells into the enteric nervous system during NEC and promotes neurogenesis, the growth of new neurons. We assessed intestinal motility in these pups by instilling methylene blue dye into the stomach, which is located at the top, 4 hours prior to sacrifice, and measuring the distance of that dye migration distally towards the cecum, which is towards the bottom of the images, at the time of sacrifice. Here we see normal dye migration in breastfed pups, significantly decreased dye migration in pups that are recovering from NEC, improved dye migration in pups that received either enteral HB-EGF or neurotransplantation, and completely normal dye migration in pups transplanted with HB-EGF overexpressing neural stem cells. Thus, HB-EGF acts as a pro-motility agent. And lastly, we looked at survival. Whereas breastfed pups had 100% survival, survival in pups exposed to NEC was significantly decreased. However, in pups exposed to NEC but treated with neural stem cells, survival was significantly improved. So the conclusions of our ENS studies were that enteric neurons and glial cells are injured during human and experimental NEC, and that in our experimental NEC model, administration of HB-EGF protects the enteric nervous system from injury, Neurotransplantation, especially with HB-EGF overexpressing neural stem cells, protects the ENS from injury. HB-EGF induces neurogenesis and acts as a promotility agent, and neurotransplantation increases survival. And based on these findings, we feel that future proposed therapies for NEC should include examination of the enteric nervous system. Our initial stem cell studies were based on the thought that stem cell engraftment into injured intestines was necessary for protection from NEC through paracrine mechanisms. In paracrine secretion, soluble proteins are secreted from granules of donor cells and affect cells in very close proximity by binding to recipient cell surface membrane receptors. Although we have documented some degree of stem cell engraftment in our studies, this didn't always correlate with intestinal protection. Therefore, our most recent NEC studies have investigated the role of exosomes in stem cell-mediated protection of the intestines. Exosomes are bilipid membrane vesicles of approximately 30 to 100 nanometers in diameter that contain microRNAs, messenger RNAs, and proteins. Exosomes are secreted by fusion of multivesicular bodies with the cell membranes of the donor cells, and they can communicate with either local or even remotely positioned recipient cells. And in addition, exosomes are ubiquitously present in blood, urine, saliva, and breast milk. So we next investigated whether exosomes could mediate the effects of stem cells in our animal model of NEC. First, we purified exosomes from neural stem cells, and we labeled those exosomes with a red fluorescent dye so that you can visualize them. We then administered the exosomes intraperitoneally in rat pups that were then exposed to necrotizing enterocolitis. Both xenon fluorescent imaging of the GI tract of the pups, as well as histologic imaging of the intestines from the pups, confirmed that the labeled exosomes did localize to NEC-injured intestine, as shown by the red staining. Here we see the effects of neural stem cell-derived exosomes on histologic injury in our NEC model. Animals that were breastfed had no NEC. In animals exposed to experimental NEC, the incidence of intestinal injury was significantly increased, and in animals exposed to NEC that were treated with exosomes, the incidence of NEC was significantly decreased. Thus, neural stem cell-derived exosomes can recapitulate the beneficial effects of neural stem cells themselves, and therefore represent a potential non-cell-based therapy for necrotizing enterocolitis in the future. So our future stem cell studies will include side-by-side comparisons to determine the best source of stem cells to protect the intestines from NEC, determination of the best route of stem cell administration, continued studies of exosome therapy for NEC, as well as utilization of exosomes as potential delivery vehicles for HB-EGF itself. Well, as you can imagine, our true long-term goal is the clinical use of HB-EGF in premature babies as a preventive or a therapeutic strategy for NEC. March 29th, 2005 was a day I clearly remember. I was sitting at my computer writing a grant on HB-EGF when the following email came across my screen. It read, "Dear Dr. Besner, I am the CEO of a privately held biotechnology company. The company's business model is to identify therapeutic candidates with promising efficacy profiles in animal models and to rapidly advance these towards clinical testing. We have for some time found various growth factors in the treatment of GI disease quite intriguing. Since we now have resources available for a new project, I thought I would contact you and inquire about the status of HB-EGF technology. We noticed that you had several granted and pending patents in the field, but we've been unable to find any commercial outfit associated with the technology. If the intellectual property rights are still available and you're interested in working with our company, I would be delighted to discuss this further. I look forward to hearing from you." Well, it turned out that this note was from the CEO of a Toronto-based biotechnology company known as Trillium Therapeutics Incorporated, and he heard back from me within minutes. Trillium was planning to use newly acquired resources to invest in one of 8 potential technologies that they were considering. On May 30th, 2005, 2 months after the initial email, I received a call from the CEO telling me that Trillium had decided to pursue the HB-EGF technology. And that was one of the happiest days of my academic career. I'll never forget that day because on May 30th, 2005, the APSA meeting was being held in Phoenix, Arizona, and I was right here at that exact time, exactly 9 years ago to the day when I received that CEO's call. After that, a licensing agreement was formed with Trillium in 2005. Production of highly purified HB-EGF suitable for human clinical administration was completed in 2007, and preclinical animal studies of HB-EGF were completed between 2007 and 2009. An additional application to the FDA for approval for use of HB-EGF in humans was filed in 2007, and FDA approval for initial phase 1 clinical trials was granted in 2010, another great day. However, while we were interested in a phase 1 clinical trial of HB-EGF in babies, the FDA demanded a much more expensive initial phase 1 trial in healthy human adults. Unfortunately, Trillium Therapeutics was a small privately held biotech company, and with the economic downturn that we faced in the past years, it just couldn't afford to continue pursuing HB-EGF technology for a small orphan disease like NEC. The day I learned that the Board of Trustees of Trillium had decided it would no longer pursue HB-EGF therapy for NEC was undoubtedly one of the worst days of my academic career. However, since then, we've never given up. We've pursued the entire avenue of stem cell therapy for NEC. We've discovered that enteric nervous system abnormalities exist in patients with NEC, which can potentially be treated with neurotransplantation, and we've devised important and likely improved potential prophylactic and therapeutic strategies to treat the disease. Hopefully, this will translate to even more efficacious clinical therapies for NEC in the future. Well, as you know, NEC is one of the most common causes of short bowel syndrome, a major clinical problem affecting newborns, children, and adults that results from extensive loss of segments of small intestine. The overall 5-year survival is 70% and is even much lower in newborns. Current treatment options are inadequate, extremely costly, and fraught with complications. A novel approach to the treatment of patients with short bowel syndrome is critically needed, and the production of tissue-engineered intestine is one alternative that we've been investigating in our lab. As with tissue engineering of other organs, the production of tissue-engineered intestine involves choosing the best scaffold material and the best cell-seeding methodology. To produce the scaffolds, we start with polyglycolic acid, or PGA biofelt, fashion it into tubes, coat the scaffolds with PLLA for strength, coat them with collagen to improve cell attachment, sterilize them, and then we use CO2 infusion to incorporate HB-EGF into the scaffolds to improve the structure of the tissue-engineered intestine that we produce. We then harvest the intestines from donor animals, digest the intestines, and use a proprietary method that we've developed in order to obtain purified crypts, since it's within those crypts that the very important intestinal stem cells and their surrounding Paneth cells reside. We then seed the scaffolds with those stem cell-containing crypts, implant them into the abdominal cavity of recipient animals. So, this is what our production strategy looks like. What we're seeing here is the donor intestine, and you can see the villus projections. Those are the villi. And in between the villi, you'll see the crypts. And in this example, we've again fluorescently labeled the stem cells in the crypts simply so that you can see where they go and how they proliferate and migrate. And those stem cells are those important stem cells that are going to go on to proliferate, to march up the crypt-villus axis, and to differentiate into all of the other intestinal epithelial cell lineages that are present in the intestinal mucosa. We then use a technique that allows us to separate the differentiated cell-containing villi from the stem cell-containing crypts, since we find that seeding of scaffolds with purified crypts leads to a much improved tissue-engineered intestine. So here's the scaffold material that we use, and the first thing that we do with it is to infuse HB-EGF directly into the scaffold material. And it ends up being released in a sustained time-release fashion. We then seed the scaffolds with the stem cell-containing crypts, implant the scaffolds intra-abdominally, and after a few weeks, tissue-engineered intestine is produced. So here you can see that the structure of our tissue-engineered intestine parallels that of native intestine, with well-developed crypts, villi, and smooth muscle layers. Immunostaining indicates the presence of all intestinal epithelial cell lineages. One novel aspect of our tissue-engineered intestine is the incorporation of HB-EGF into the scaffold material. Here we see tissue-engineered intestine produced in non-HB-EGF-coated scaffolds on the left and in HB-EGF-coated scaffolds on the right. The tissue-engineered intestine produced in the HB-EGF-coated scaffolds has more well-developed smooth muscle layers, increased numbers of crypts, and longer villi. Here we see histologic images of tissue-engineered intestine produced from scaffolds infused with increasing concentrations of HB-EGF. And we found that the higher levels of HB-EGF that we infuse into the scaffolds, the longer the villi that are produced, even exceeding the villus length of native intestine. So our ongoing work in this area involves optimizing the scaffold material, the cell seeding techniques, and the growth factor infusion methods for the production of tissue-engineered intestine, and to use these optimized parameters to produce human tissue-engineered intestine. Well, I wanted to end my talk by bringing to your attention something that has me quite concerned lately. As you all know, researchers are currently faced with the major challenge of obtaining research funding. The NIH budget doubled between 1998 and 2003, leading to significant breakthroughs in research, including sequencing of the human genome. However, NIH funding has remained flat over the past years, making it difficult for even seasoned investigators to obtain new grants or to retain previously funded grants. Overall, less than 1 in 5 grants is guaranteed funding, and at times are only partially funded after long delays in resubmissions. The NIH has a type of grant submission known as the K Award, a 5-year mentored award targeted for young investigators. And it's typically the young investigator's first foot into the door of the NIH. This graph shows the success rates for K08 basic science grants between 2004 and 2013. In red, we see that the success rates for funding have been stable at a remarkable 40%, the highest success rate for any type of NIH award. However, the number of grants funded has significantly decreased as shown in blue, and this is a direct result of decreased numbers of K08 grants being submitted as shown in brown. This next graph shows the same analysis, but this time for K23 clinical research grants. Again, the success rate remains the same at about 40%, but the number of submissions has declined. I believe that this is because young investigators have become discouraged by what they perceive as something that's almost impossible to obtain. Furthermore, K awards are relatively small, only bringing in about $75,000 a year for salary and $30,000 to $50,000 a year for supplies over the 5-year period of the grant. And they require 75% of the faculty members' time to be dedicated to their research training. Sadly, many department heads are actually upset when one of their faculty members obtains a K award because it translates to decreased revenue generation to the department, since the young surgeon would have brought in more money spending their time in the operating room. We need to rethink this. Pediatric surgeons have contributed immeasurably to scientific discoveries. Many sitting in this room have made seminal discoveries crucial to the care of our pediatric surgical patients. All of these investigators had to start somewhere, and we need to do everything that we can to mentor and foster the ability of young, upcoming physician scientists who will eventually continue pediatric surgical research in the future. This interesting, recently published paper investigated the success rates for pediatric surgeons in obtaining NIH grants. Current APSA members were cross-referenced with the public records of NIH grants funded between 1988 and 2012. 83 pediatric surgeons received 171 independent NIH grants, representing 13% of APSA membership. 6% of APSA members currently have active NIH funding. There were 28 K awards during this period, And 39% of K awardees subsequently went on to receive R01 grant funding. This is big research funding now, with 63% of K awardees transitioning to some type of independent NIH award, which is really quite an excellent number. However, with the number of K awards decreasing due to fewer submissions, I fear that these numbers may worsen over the next 5 to 10 years unless we continue to do everything that we can to support young pediatric surgeons who are interested in research. There's immense joy in helping others become the academic pediatric surgeons of the future. I believe that successful mentoring of our younger trainees is the most important legacy that we can leave as pediatric surgeons. Doing so will have the greatest likelihood of influencing the care of children in the future. Mentoring needs to be a crucial responsibility of the American Pediatric Surgical Association as an investment in our future. I cannot imagine any pediatric surgeon who exemplifies a better mentor than Dr. J. Grosfeld, after whom this lectureship is named. It's a distinct honor to have had the opportunity to give this lecture in his name, one for which I'm immensely grateful and for which I'll certainly never forget. I would like to particularly acknowledge some of the individuals who've supported my career along the way. I'm deeply indebted to Dr. Folkman for taking a surgical resident with little research experience into his lab. While it is possible that a love for research is in the genes, as my mother would have said, if we don't encourage young investigators to try research and to support their early careers, they may never realize this passion. In 1990, Dr. Bill Clatworthy was our visiting professor during my pediatric surgery training, and I believe that that was instrumental in my eventually locating to Nationwide Children's Hospital after I finished my training. Dr. Clatworthy welcomed us to Columbus and to his home, and I feel incredibly honored to hold an endowed professorship in his name. I will always be indebted to Don Cooney, my chief when I was a pediatric surgery fellow. Dr. Cooney, myself, and my husband relocated to Columbus upon completion of my training, and Dr. Cooney provided us with the time and resources to establish the Pediatric Surgery Research Laboratory in Columbus, which has persisted with continued NIH funding for the past 2 decades. Dr. Donna Caniano supported my ability to continue my research during her time as chief, and I thank her for that. And I'm very grateful to Dr. Larry Moss for believing in me and for supporting me as the chief of pediatric surgery at Nationwide Children's Hospital. I'm particularly indebted to each and every one of my partners in pediatric surgery at Nationwide Children's who have valued my research endeavors over the years. These are the research fellows who over the years have accumulated all of the data that I showed you today, for which I'm extremely grateful. One of the greatest pleasures of my career is to have watched these fellows grow and develop in the lab. Dr. Zhao, Yang, Watkins, and Wei produced the stem cell data that I showed you today, and Drs. Liu, Boomer, and Rager produced the tissue-engineered intestine data. Although the next 2 slides are the last that I'll show you, they're the most important. Not only did I discover HB-EGF during my time in the Folkman lab, but I also discovered my husband, Dr. David Brigstock, who had traveled from England to do a postdoctoral fellowship in the lab. Discovering HB-EGF was quite good, but discovering David was truly great. He's been there for me in good times and bad, and for that I'm truly grateful. And I just want you to know that he has an NIH grant due in 5 days, but he's still sitting in the front row to hear this talk. And lastly, my amazing children, Matthew, who's 19 and studying political science at George Washington University, and Nicole, who's 17 and starting her senior year in high school. They've taught me more about life in general, justice, and human rights than I ever could have imagined. In closing, I think you'll all agree that we are incredibly fortunate to have the jobs that we have. Every day we go to work with the opportunity to solve new research questions in the laboratory in order to improve the health of our patients, to continually learn new things and to advance our surgical capabilities, and to save the lives of the children that we care for today and in the future. ABSA must continue to serve as the main structure which helps all of its members, especially its youngest members, in achieving their academic and professional goals. Thank you very much for your attention.